Hi, and welcome back to the Applications Lab of Dynamic Biosensors. I'm Nena Macheco, and I'm going to guide you today through a molecular interactions tutorial of a DNA binding protein. Taking advantage of the multi-channel optical detection system of the new heliX biosensor, we are going to multiplex several binding reactions into one measurement. This allows to screen 4 binding partners of interest within 20 minutes. The increased sample throughput reduces the measurement time on the heliX by 75%.
DNA and RNA binding proteins are an intriguing class of molecules with many functions essential for DNA replication and for gene regulation and expression. To fulfill any of these functions, the first step for these proteins is to bind to DNA or RNA. In other words, their binding affinity is a key component of their activity. Many nucleic acid binding proteins have high target specificity. Target specificity and binding affinity may be impacted by secondary structure as well as nucleic acid sequence. Would you like to know more about measuring the specificity and binding efficiency of molecules to DNA or RNA? The following tutorial measures the interaction between an RNA polymerase and its promoter DNA. RNA polymerases have received a lot of attention recently due to the COVID-19 pandemic, since the virus uses an RNA polymerase for replication. However, we will use here the simpler model of the bacteriophage T7 RNA polymerase. Before we get into the technical assay setup, let's start with a basic understanding of the function of the T7 RNA polymerase. RNA polymerases bind to a specific DNA sequence, the promoter, as a first step in the transcription of DNA to RNA. The T7 promoter has been defined as a region that includes just 18 base pairs. The activity of the T7 RNA polymerase is to transcribe single-stranded RNA from the template DNA strand. The first phase of RNA transcription is the association of the polymerase to this double-stranded DNA target sequence. Right after association, the polymerase unwinds the end of this double-stranded DNA and remains attached to the now single-stranded template strand. If it is supplied with the right nucleotides, it can now initiate the synthesis of RNA. Since the first step, the association, is a crucial rate-limiting step that regulates transcription efficiency of the polymerase, it is often targeted for regulating the expression level during recombinant protein expression or for in vitro RNA transcription. Let's take a closer look at the regulation by varying the promoter sequence. Previous studies on T7 RNA polymerase binding to its promoter have narrowed down the core sequence for binding and transcription initiation to the DNA sequence shown here. Interestingly, effects on affinity could be observed for changes within, but also both up and downstream of this core region. Previous studies have shown the effect on RNA transcription when introducing mismatches in the promoter sequence, when deleting up or downstream regions, or when exchanging base pairs. However, if we think of the multi-step process required to transcribe RNA, we do not know if binding, unwinding, or any other step of transcription is affected. We need to look at the binding isolated from the downstream events to determine the effect of promoter composition on binding affinity. An increase in binding affinity may be caused by faster association or by slower dissociation of the polymerase DNA complex. To understand the underlying mechanism for a change in affinity, it is necessary to resolve the binding kinetics. The association rate for the T7 polymerase and its promoter can be measured and is described by the association rate constant K-on or Ka. Once bound, the polymerase DNA complex can separate at a rate quantified by the dissociation rate constant K-off or Kd. The equilibrium dissociation constant, Kd, for the interaction is defined as the ratio of K-on and K-off and is the concentration at which 50% of the analyte is free in solution and 50% is associated as a complex. If you are not fully familiar yet with the theory of binding kinetics, you can find a more detailed explanation in the Basic Kinetics tutorial linked in the info box. T7 polymerase has very specific contact points with its promoter DNA. Changing the DNA nucleotide composition can affect both the association and dissociation rates. Can switch sense measure changes in association and dissociation rates when the promoter sequence is altered?
Absolutely. Let's run a fast experiment to see if the observed effects on the binding rates when changing the promoter sequence can be confirmed. For this purpose, let's pick one sequence of each type of modification in addition to the consensus promoter sequence. The consensus core target sequence is flanked with upstream and downstream regions and serves as our reference and positive control for the T7 RNA polymerase batch used. We will include a sequence with a 2-nucleotide mismatch in the Tata box, as well as a sequence lacking the Tata box and downstream template completely. Thereby, we can separate binding affinity from unwinding of the template, the second step of the polymerase activity. Of course, with a sequence-specific DNA binder, you would like to ensure that in your chosen conditions there is no off-target binding taking place. Specificity can be crucial in medical applications such as CRISPR-Cas9 targeted gene therapy. The real-time control of non-specific binding to a scrambled DNA sequence will give us full control and maximum data from this quick experiment. This sounds like many measurements to you? Actually, it is pretty fast and easy to screen these sequences with the help of the SwitchSense technology. Let's take a look at what we need to perform this kinetic experiment. First of all, we need the samples that we would like to measure. I have a kit here containing the ligands and the analyte T7 for our experiment. Second, we of course need a measurement device to record the interaction. We are going to work with this heliX Plus device. The samples need to be transported through the microfluidic system with running buffer. For this purpose, I have placed a bottle with buffer in a physiological salt concentration range into the device. The interaction itself is taking place on a sensing surface. Therefore, we have to insert a blank biochip that can be functionalized with our samples.
The heliX adapter chip features a versatile linker system to immobilize and exchange molecules on 2 detection spots located in the same flow channel. The linker system consists of 3 building blocks, which are all made up of single-stranded DNA. The anchor is a 48 nucleotide DNA strand that is covalently attached to the gold microelectrodes of the chip via thiol chemistry. The chip comes pre-modified with anchors, which remain on the surface at all times. The 2 detection spots are modified with different, unique anchor sequences so they can be individually modified later by a technique called DNA-encoded addressing. The second element is the adapter strand, shown here in black. The adapter is a 96 nucleotide DNA strand, whose lower half hybridizes to the anchor strand. The top end of the strand is labeled with a green or red fluorescent dye for detection purposes. The adapter strand is exchangeable and allows our assays to be very flexible. We can choose different fluorescent labels, different lengths of adapter strand, and we can easily regenerate the surface again and again. We can even integrate sophisticated DNA nanostructures such as DNA origamis. The third element is the ligand strand, which is modified with the molecule of interest. The ligand can be anything: a protein, a peptide, a small molecule, or even an RNA or DNA sequence. In our case, the ligand is the double-stranded T7 promoter sequence and the 3 variants described earlier. Each of the 4 promoter variants can be synthesized as an overhang of the ligand strand. The heliX biosensor system can be used to measure 4 interactions simultaneously on a single heliX chip. This is achieved by using adapter strands labeled with either a red or green fluorophore and that are targeted to either the first or second electrode. Red and green fluorescent signals are detected by single photon counters at each of the 2 electrodes. The second electrode may be used for real-time referencing or, as in the case of today's experiment, for increased multiplexing. The heliX features 2 measurement modes, static and dynamic. Static mode is primarily used for real-time binding measurements, while dynamic mode is used primarily to analyze ligand size and conformation by measuring their hydrodynamic friction. For measurement of T7 RNA polymerase binding to its DNA target, we chose static mode. The mechanism for static mode sensing is quite simple. The fluorescent dye attached to the top of the adapter strand is a sensitive reporter of changes in its chemical environment. The dye and the analyte ligand complex are both subject to Brownian motion, causing them to tilt, rotate, and frequently collide with each other. In the presence of analyte molecules, the fluorescence emission intensity of the dye changes by collisional and other quenching processes. The signal decreases when the analyte is bound to the ligand and increases again when the analyte-ligand complex dissociates. Whether the signal increases or decreases upon analyte binding is actually not important. What is important is that the signal change always scales with the fraction of analyte molecules bound to the sensor surface. To multiplex the interaction analysis of an analyte with multiple ligands, we immobilize the ligands simultaneously on our surface. The DNA-based heliX adapter biochips allow full flexibility and control of ligand immobilization. If you are working with two different targets, you can color code them by hybridizing one of them to a red-labeled adapter and the other one to a green-labeled adapter. These ligand strands are then mixed in one sample vial. Since the anchor DNA at each electrode on the heliX adapter biochip is the same for both colors, the ratio will be transferred to the desired electrode when your ligand solution is hybridizing. Due to the color code, the multi-channel optics of the heliX will record the interaction taking place with each ligand individually. Let us show you the advantage of multiplexing by going through a calculation of runtime. Based on the interaction settings for the tutorial, measuring the interaction of an analyte with 4 different ligands sequentially would take about 84 minutes. Not too bad, but this time can be reduced by half by functionalizing measurement spot 1 with 2 color-coded ligands simultaneously. But of course the adapter chip has 2 measurement spots. Can those also be functionalized with ligand? Yes. So let's take advantage of that and multiplex the surface functionalization on spot 2 as well. This leads to 4 different ligands on the sensor surface which can interact simultaneously with the analyte in solution. And from one 21-minute kinetic experiment, we are able to measure 4 individual binding interactions and reduce runtime by 75%.
Since multiplexing is saving us so much time, we will directly apply it in this tutorial. We can run the multiplexing experiment following the standard switch sense workflow. We always start with the immobilization of our ligand or ligands on the fresh sensor surface. In our case, we would like to immobilize 4 different double-stranded DNA oligos as overhangs of the nanolevers. These ligands will be our targets of interest. After the surface is functionalized, we want to flush in the analyte, the T7 RNA polymerase. This will be the association phase of the kinetics. By presenting the DNA oligos on the surface, we allow the DNA binding protein to freely move in solution without the need for any modification or restriction of conformational changes while binding. The association phase is ended by the injection of running buffer, and while that is flushing over the sensor, we can observe the dissociation of the polymerase from the DNA. With that experimental plan in mind, I will now show you how to easily set up your planned multiplex assay in the Helios software. I will first go to Assays and open up a new empty assay. After entering the name, I can start adding method elements of my choice. For the screening of interaction kinetics, I will choose the fast kinetics in the static mode. This method includes the entire workflow of ligand immobilization, analyte association, and dissociation. The parameter list provides the heliX with all the information it needs to run my kinetic experiment and analyze it afterwards. Importantly, for a multiplexing assay, I will go to the details of the ligand and provide all names and the respective measurement spot and color code. Now I have finished my assay setup and would like to know which samples I actually need. The software is providing me with a list of all required samples in the autosampler tray overview. The multiplex DNA binder kit contains the ligand DNA mix and the analyte T7 polymerase. I have to make sure to put them into the indicated locations, or I could modify the sample positions by drag and drop in the software. I'm inserting the 96 well plate with the analyte into the autosampler tray first. The small vial contains all 4 target DNA oligos. The 4 different ligands have been pre-incubated individually with their respective adapter strands to ensure the specificity of the spatial color code. In the kit, I received the ready-made mix of all 4 in one vial, ready for the multiplex functionalization. I can directly transfer the provided vial into the autosampler. Once the assay is running, we can see the data collection during a measurement in real time in heliX. All data recorded is automatically saved to the database and is ready for analysis when the assay has finished. This tutorial measurement finishes after just 21 minutes. To analyze the multiplexed assay, we open the list of experiments. You can use the search function if many experiments have been performed already and your multiplexed assay is not on top of the list anymore. I open the experiment by double-clicking on it. Then I can start the individual one-to-one kinetics analysis with the big blue Analyze button. And this is the finished analysis. heliX is providing us with an overview of the recorded data as well as tables of the fitted association and dissociation rates and affinity constants. The decrease of fluorescence from the initial baseline during the association phase indicates the quenching of the sensing fluorophore by T7 binding to the ligand. This fluorescence recovers during the dissociation phase if the protein DNA complex is falling apart and the T7 is being washed away. We can immediately see from the plot that all 4 targets interacted quite differently with the analyte T7. The modifications of the promoter had a big influence on the binding kinetics.
The multiplex experiment described in this tutorial is designed as a screening experiment. Nevertheless, we are able to begin to understand the kinetics and affinities for the binding interactions. There was no interaction between the T7 RNA polymerase and the scrambled DNA sequence. This is a useful real-time control of binding specificity, which confirms the high sequence specificity of the T7 polymerase for its consensus promoter. The polymerase showed good interaction with the consensus promoter sequence with a KD in the 2-digit nanomolar range. Interestingly, the affinity increased when the TA mismatch was introduced. The association rate constant increased by a factor of 2, and the dissociation rate decreased also by a factor of 2, resulting in a KD of around 5 nanomolar. Affinity was increased further when the truncated promoter missing the transcription initiation site was tested. In this case, the on rate increased 10-fold while the off rate decreased 30-fold, resulting in a 2-digit picomolar KD. We should be aware that the dissociation was slowed down so much that the signal change observed during the screening experiment might have been insufficient to calculate an accurate curve fit. We can therefore use the preliminary binding data generated in this screen to help establish the conditions for a higher precision study conducted with multiple analyte concentrations. We will now show an example experiment for the more in-depth kinetics analysis. The TA mismatch promoter showed good interaction in the multiplex screen, so we will use it as the ligand and a series of T7 concentrations as analytes in order to demonstrate how to more accurately measure binding kinetics and affinities. You can see in the data plot the concentration-dependent dissociation of T7 to the mismatched DNA. As expected, higher analyte concentrations result in larger signal amplitudes until the amplitude reaches a maximum at full surface saturation. The rates shown were calculated by a global fitting algorithm which finds the best fit for all concentrations together, in contrast to the individual fits applied in the screen. The replicates of the same analyte at different concentrations improve the accuracy of global curve fitting and therefore result in more accurate KD data. Similar studies were conducted for all T7 promoter variants. The summary of the binding affinities determined in this switch sense tutorial illustrate the broad range of affinities that can be covered. The increase in affinity may seem surprising at first, but our results are supported by other studies reported in the literature. For the binding of the T7 RNA polymerase to its consensus promoter, Tang et al. Reported KD values in the 50 nanomolar range, while Banwar and Patel reported values in the 150 to 480 nanomolar range. The absolute value is strongly influenced by the measurement conditions and protein source. Therefore, we need to look at the relative change of affinity when modifying the promoter. Banwar and Patel saw increased affinity when they introduced single mismatches in the melting region, with KD values in the 290 to 950 picomolar range. They saw even stronger binding when a 4-nucleotide bulge was introduced, with KD decreasing to 2.5 picomolar. This fits very well with our switch sense results of an increase in affinity from 2-digit nanomolar with the consensus promoter to low nanomolar and even picomolar affinity when modifying the melting region. As described earlier, RNA transcription includes several key steps which must be balanced for efficiency. The initial step is binding, but that must be followed by melting of the DNA strands and movement of the T7 polymerase along the DNA in order to produce the RNA transcript. According to Banwar and Patel, the melting step is driven by energy released from the binding step. Therefore, already unwound DNA, or removal of the Tata box, conserves this binding energy and results in higher affinity. We have shown the effects of several changes to the T7 promoter on binding kinetics. While it is required for promoter activity that T7 RNA polymerase bind its promoter sequence, it is apparent that binding that is either too weak or too strong can negatively impact transcription. In more complicated systems where promoter activity requires the presence of one or more transcription factors, it may be important to look at binding of multiple proteins to fully understand the effect of DNA sequence changes on transcription. SwitchSense technology can help develop an understanding of complex systems and help predict the effects of modification to one or more binding partners. As we have demonstrated, SwitchSense is the ideal solution for measuring interactions between nucleic acid binding proteins and their targets. Targets may be easily multiplexed to achieve substantial reductions in experimental time. The heliX chip can be rapidly regenerated with as little as 2 picomoles fresh sample for each measurement for highly reproducible results. The broad dynamic range of the SwitchSense system enables measurement of both very weak and very strong binding interactions, with KD values ranging from 1 millimolar to 50 femtomolar. The heliX instrument is easy to use with simple workflows and powerful analytical software. This is the ideal technology to measure binding kinetics of your nucleic acid binding protein.
All right, that was all for today. It was my pleasure taking you through this multiplexing tutorial. Please do not hesitate to contact us if you have any questions or would like to repeat this measurement by yourself. If you're interested in this kind of content, don't forget to subscribe to our YouTube channel and make sure to also view our other tutorials. Ciao!